Okay, I think we started with Lamarckism, right?
Yes, sir.
Okay, Minashi, were you there in the last class?
No, sir.
Oh, you missed the last class? Okay. Miraj, what about you?
Me too, sir.
Okay, then I think it's Nabil who's the only consistent person apart from me here. Okay, Nabil, so would you like to give a quick summary to your fellow batchmates to help them orient where we are and what we were doing?
Yes, sir.
Yes, please. Try to explain, okay? Just explain a little bit. Don't just go by the term so that they can understand the contents.
We discussed about Lamarckism. So this was a theory given by a person called Lamarck. So he was essentially before Darwin. He was another person who went against the so-called theory of special creation, and he had his own concept of evolution. That although we know that Darwin said that it is because of the chances that occur in nature, but according to Lamarck, it's because of desire. Like if a person wants to be taller, eventually he will lead to a new species that will be taller. So
Yeah,
one such
but
example.
for that some actions have to be done also. Okay, so doctrine of desire is not just desire, but certain actions that we take. Like giraffes
Yes,
started stretching their necks,
yes,
and a person who wants to be taller will start hanging, or
yes.
stretching the body. Yes, continue.
So what he said to prove his theory, he gave the example of a giraffe. He told that as food got scarce, only the leaves on the trees were a short source of food. So slowly horses started to extend their necks, and after a long period of time, they evolved into giraffes. However, many people still debate on this. Although some part of it is still debated to be maybe true, but mostly it is not believed to be. The next one is mechanism of evolution. So main mechanism of evolution is mutations. So sudden, random, non-directed, and inheritable changes in the DNA. So these are the factors. These were the factors given by Mendel. So Mendel said that there are some factors which are passed on from parents to offspring, which we know as alleles. So and this concept of mutations was given by uh, uh, Hugo de Vries. Then mutations are random and directionless, whereas Darwinian evolution is non-random and has direction. So as we know, mutations during reproduction, there's a random chance that maybe some part of the DNA might get changed. So it's random, but according to Darwin, it has direction. So if
Yeah.
you specifically eat insects, your progeny will be more developed to eat insects. Then,
Yes.
then Hugo de Vries believed that mutations can lead to larger changes, causing speciations in short time frame, as short as one generation. So he believed that instead of it being like a Darwin's theory, where over his long generations, slowly, there's a change. Suddenly, in one generation or in two generations, there might be a new change coming up in the species. Then he also believed that a mutation causes speciation, and this is known as saltation. That is a single one-step evolution. Then we discussed about allele frequency or gene pool or gene pool being finite. So this was given by it's also known as Hardy-Weinberg principle or the principle of genetic equilibrium.
Yes.
So principle states that allele frequencies in a given population are always stable and remain constant from generation to generation. That is where the gene pool stands for the total genes and their alleles remains constant in the population. And the sum of all allelic frequencies is 1. So what it basically means is that every person is having different combination of genes, but all of those combinations of genes are not like newly made on the spot. Those combinations are all selected from an already existing gene pool. So if there is any change in that gene pool, meaning the introduction of new genes, then it will be detected as a change in the equilibrium of Hardy-Weinberg principle. So they gave an equation where p square is plus 2pq plus q square is equal to 1. Here, this is similar to the equation of a square plus 2ab plus b square is equal to a plus b the whole square. So we have p plus q the whole square is equal to 1. So there are factors that can affect gene flow. First one is gene migration or gene flow.
Yes. So
Uh,
and also Nabil, what is this P and Q basically, and
it
why?
is in the case of diploid organisms, we know that there are two alleles for a particular gene. So like either one of the two options. So if we consider the case of tallness, there are two options. The person might be either tall, might have the chromosomes for both for tallness, both for dwarfness, or one for tallness and one for dwarfness. So by using permutations, combinations, if we represent tallness characters frequency as P and shortness as Q, then you will have for tallness, the chances will be P square, shortness, it will be Q square, and for uh, like having both the chromosomes,
Yeah. So
it will
basically, it's
be,
the frequency of the allele, right? The
yes, sir,
frequency of the allele to be present in any population. Now,
yes, sir.
frequency of both similar kind of alleles being present is P square or Q square, depending on two different alleles. And frequency of a heterozygous for, condition is 2PQ, like I have shown. Yes, continue.
Then next we discuss the 5 factors that can affect the Hardy-Weinberg principle. First one is gene migration or gene flow. So if a section of population goes from one place to another to mix with the pre-existing population in that region, then this will lead to gene frequency changes in the original population as well as new population. So due to this gene migration, new genes or alleles will be introduced to new population and some are lost from the older population. And gene migration happening multiple times is known as gene flow. Then second one is genetic drift. Sometimes the same change in an allele frequency, like gene flow, happens suddenly and by chance, like an avalanche, or like two populations of beetles that live together. Suddenly, one got stopped by an elephant, and there'll be sudden change in the gene pool of that particular species that will lead to change in the Hardy-Weinberg principle. Then 3, 4, and 5 factors are mutations, genetic recombinations, and natural selections. So some of the important questions that can come are the list the 5 factors that can affect the Hardy-Weinberg equation or equilibrium. Then next we also discussed about founder effect. Sometimes the change in an allele frequency is so different that the new species formed is formed from the new population. So the originally drifted population becomes a founder. This is like if one species gets suddenly there's a mutation and it results in a whole new progeny that's very different from the older generation, then the older generation gets considered as a founder, and this effect is known as a founder's effect.
Yes, right. Thank you so much, Nabil. Indeed, a very, very well explained summary.
Thank you,
Okay,
sir.
so yeah, very good. So we have also been joined by Abia. Abia, how are you? Good evening.
Good evening, sir. I'm good.
Were you present in the last class?
Yes, sir.
Okay, very good. Then I think, and others, did you understand what Nabil just summarized? At least some gist of it, right?
Yes, sir.
Okay, very good. So in today's class, we'll just take this conversation, this discussion forward before going to the evolution in plants and animals. So mutations, or as I told you, that mutations most of the times, so Nabil also said this, mostly mutations are deleterious, right? And as we have, because we have already discussed it, Nabil, will you tell why mostly mutations are deleterious or damaging?
Because uh, if something random happens to, like we've evolved to be particularly suited, and our body functions are all particularly suited to what we need. So if there's a sudden change, there's a high chance that it might go wrong and might affect the person's overall physiology and development. So mostly they are considered to be deleterious. However, in a very small chance, it is beneficial to the organism.
Yes, so it's just a random chance that something it might have incorporated somewhere where this mutation is advantageous. Now, these advantageous mutations, how do they actually help the organism is they can cause speciation. So the concept that I'm going to start with today is the concept of speciation. You know what is speciation?
Splitting of like division of species, branching of species to new ones.
Sorry, come again. Nabil,
Branching
your
of
voice.
a particular species to create new species.
Did you say branching?
So like one
Okay.
species will
Mhm.
split.
Okay, so in simple, if I understand you correctly, and if I can say it like this, can I say that you mean that in simple terms, it is a formation of new species from the pre-existing ones, right?
Yes, sir.
Yeah, okay. So exactly that. So write down speciation. Speciation is the process. Speciation is the process of emerging. Speciation is the process emerging of new species. Species. The singular is species, plural is species. So emerging of new species from pre-existing ones. Through evolution of new and different phenotypes or characteristics.
Sir, I have one question, sir.
Yes,
Species are
please.
those organisms that can breed with each other, right?
That can?
Breed with each other.
Yes,
Like they won't be able to breed with any other organism, only with those particular species. So since
yeah,
lion and tiger are different species, how did we get tigers?
exactly. Good question. So the definition which you said is not complete. So definition says that species is defined as a group of organisms which are capable of breeding or reproducing with each other under natural conditions. So in nature, their instinct behavior is to go for the female. So males, let's say tiger male, will not go for a female lion in nature. Okay, because there is behavioral evolution also, and in natural conditions, their compatibility, the kind of behavior that they show, that only allows them to reproduce within the same species under natural conditions. But under artificial conditions, where you can just do an IVF and put it back into the tiger. So there are many ways to create a tiger. You take a tiger sperm or a lion sperm and a tigress or a lioness ovum, do a IVF, and then do a intra-fallopian transfer or intrauterine transfer. You understand? That's one way.
Yes, sir.
Or under artificial conditions where they are bred in captivity, so they don't know that naturally with whom they should breed, right? So if a tiger male and a lion female is bred in captivity and they are the only male and female in an enclosure, so they can be trained to get rid of their uh, natural instincts. You understand? For that matter, the natural instinct of a tiger is to hunt, but haven't you seen tigers which are tame and used in circus? Earlier used to be in circuses, and you can even tame it as a pet.
Yes, sir.
So now you understand?
Yes, sir. And one more question, sir. Like
Yes.
our body has evolved to have some microbes in our intestines for helping in digestion, right? Like
Exactly.
lactobacillus. Then why don't
Yes.
we have bacteriophages for protecting against bacterial infections?
Why don't we have bacteriophages to protect against bacterial infections? Yes. Amazing. Beautiful question. Now,
Just similarly for like treatment, instead of taking antibiotics, why don't we just inject like bacteriophages?
yes, very good question. Now, I might not be able to give you all the aspects of the answer here in class, because some of them required some high advanced concepts for you to understand. But if you see it like that, bacteria can just, it's not like we don't have viruses in our body living, but mostly viruses do not do anything. Like they cannot be useful to us as organisms because they don't have a cytoplasm and they are not producing something which we can use for our benefit in their cytoplasm, right? They are just hijackers. So they are not like a pathogen or a parasite, which can also come under a symbiotic relationship. So to make a symbiotic relationship, basically you are talking that over millions of years of evolution, why we could not make a symbiotic relationship with viruses, right?
Yes, sir.
Yeah. Okay. Because viruses, I only infect. Okay. They are just hijackers. So if they recognize a human cell like COVID, SARS-CoV-2 does, then it will infect, enter the cell, make its own copies and kill the cell. That's all it does. It just needs, it can only do one thing. It can self-replicate itself. It does not synthesizes within itself anything that is useful to us. Do you understand? Now,
Yes, sir.
they are killing bacterial cells the same way they are killing a human cell. The viruses which can recognize bacterial cells cannot recognize human cells. And the ones which can recognize human cells cannot recognize bacterial cells. Now, to make a symbiotic relationship with us so that they can help us getting rid of infections, we need to, harbor them inside our body, right?
Yes, sir.
We need to provide them conditions to grow and multiply. Am I correct?
Yes, sir.
And they can only grow and multiply within a cell by infecting the cell. Is this clear?
Yes, sir.
Which means they can only create infections. And if we allow them to create infections, we cannot allow a virus which can kill bacteria to proliferate within our body. You understand why? Because they will not recognize our cells as host. So that's one problem. Is it clear to you, Nabil, or not? Is it clear to others?
Yes, sir.
Yeah. So that's one part of the problem. The other part of the problem is that they cannot, what you are correct, that instead of taking antibiotics, and in fact, it is a emerging field of research. So actually you are thinking like a researcher only, a young researcher, because I have worked with a professor who is actually exactly interested in this thing. She believes that phage therapy, it's called phage therapy, bacteriophage therapy. Okay, Nabil. So I'm sure that you might feel a little happy knowing this, but it is actually in reality, people are doing the same. They're working on the same idea that you are putting forth. That can we utilize phages, bacteriophages, whenever we get a bacterial infection. So if I have bacterial infection, instead of taking antibiotics to which they might have resistance, I can use bacteriophages that might kill those bacteria as an injection in that organ or in the blood. Do you understand, Nabil? So it's there.
Yes.
But this therapy or this field is still progressing. It's not completely developed to be used therapeutically. In some pockets of the world, these things are being tested and they are proven to show some improvement and effect, especially for tuberculosis. People are trying to figure this out because the bacteria for tuberculosis, it's a mycobacteria, right? It's very difficult to kill. and it has developed resistance against many drugs that we use to treat TB. You understand, Nabil? Is it clear?
Yes, sir.
Okay. Good question. Anything else? Did,
Sir, one more.
did it answer your question?
Yes, sir.
Okay. Yes. What's the other
One
one?
more good question, sir.
Yeah.
In our brain, the main reason we're having those wrinkles, right, is so that we can increase the surface area for neurons, right, sir? Like, so that we can pack more
Oh,
neurons into small space.
you're
Right?
talking about the gyri of the brain, like the
Yes,
structure of the
sir.
brain. Okay.
Yes, sir. The wrinkle ones. Sir, why don't we just make it like spherical? Because in those gaps, you can fit many more neurons.
Oh, I see. See, it's very simple. I don't even have to go to neuroscience to explain you that. This is one circle, right?
Yes, sir.
Yeah. So first thing, these are not just to pack neurons. One thing, these folds are not just to pack neurons like we think. So of course, if this is a circle, you can pack a lot of neurons. But neurons are not just packed; they are arranged in the brain, right? They are arranged in a way where the top layer has the cells and the axons and the dendrites goes down like this. You understand? So you need
Yes.
more. The more the surface area, the better. The more number of neurons can be functional. If it is just about packing, you can just put it in a bag and pack it any orientation. But if all the cell bodies have to be in one orientation, you now are restricted by just the outer surface.
Yes, sir.
So if all the neuron cell bodies have to be there only, so there is a limit how many cell bodies can be here in a circle, right?
Yes, sir.
So, but if instead of a circle I make it like this, then within the same space there can be many cell bodies. Understand?
Yes, sir.
Simple. Okay.
Yes, sir.
Cool. Anything else, Bill?
I think-
Anything else? Shall I move forward?
Yes, sir. Yes, sir.
Okay. So speciation is clear, right? Now write down. Write down. Microbial experiments have shown. Microbial experiments have shown experiments have shown that pre-existing advantageous mutations. So if any mutation is advantageous, so these pre-existing advantageous mutations, when they are passed on to next generation after next generation, they get selected in nature and they create new phenotypes. You understand? It's simple, right? It's already creating, for example, let's say there is a mutation in a bacteria that can now help the bacteria get antibiotic resistance. We were just talking about that, right? Everyone say yes or no?
Yes, sir.
Right? Now, if
Yes, sir.
any mutation is giving the bacteria antibiotic resistance, it must be through some mechanism, right? Let's say the bacteria had a mutation in a protein which now can act as a a pump to throw antibiotics out even if they enter the cell, and this is exactly how many bacteria get antibiotic resistance. So what they do, they realize that something is coming inside my cell which is killing me, so they cannot some antibiotics which enter the cell and then work. So for these antibiotics there is a efflux pump. So these antibiotics enter and then there is a pump like a channel that keeps throwing it out of the cell again. So they have identified that this is the harmful thing. It's just like if something harmful comes into your house, what do? Throw it out, right?
Get out.
Clean it. Yes. So they do the same thing. but it has happened through mutation. So over time, there will be new phenotypes created. And this species of bacteria, and this group of bacteria will become a new species, different from the species in which the mutation happened. Is it clear?
Yes, sir.
So there is actual microbial experimental proof of speciation. It is not just a concept that we talk about, because someone can ask, no, has anyone seen speciation happening? Yes, if you can see it happening in a microbial system, which is, faster system. And I told you in previous classes that microbial or unicellular systems evolve faster. Yes or no?
Yes, sir.
Why they evolve faster?
Because they only have to be concerned about one cell instead of our body
Yes,
where we have to
they're,
be concerned about like millions
yes,
of cells.
yes. And basically their lifespan and life cycle is very short. For a E. coli, it is just 20 minutes. That is the most important reason. Okay.
Yes, sir.
Okay. So write down. What did I say? Microbial experiments have shown that pre-existing advantageous mutations are selected in nature and give rise to are selected in nature and give rise to new phenotypes. Okay, and these new phenotypes over a few generations result in speciation. So do you understand now that some members of the population now, after few generations, so one member got some mutation, it was useful, and it got selected because whenever it is that member is reproducing, it is producing progenies that are better than the rest. Yes or no?
Yes, sir.
So they will keep getting selected, and over time, after a few generations, they will become a new species, different from the previous one in function and phenotype both. Okay. Now write down just one more point: natural selection. Have we defined natural selection already? If not, let's just do it once more. Natural selection. Write down. Natural selection is a process in which heritable variations. What is heritable variations?
Which can be passed on from parents to progeny.
Yes. Heritable, variations. Which enables better survival. Which enables better survival uh, which enables better survival are made to reproduce more in nature and leave greater number of progenies. And leave greater number of progenies. Okay. Is it clear? So natural selection basically operates through survival of the fittest. So whoever is fittest, fitting the best in the environment, gets selected and survive the most. Make sense to all of you?
Yes, sir.
Okay, very good. Now, having known all the mechanisms and everything, before I go, so the next part we will do a in-class assignment now. Okay, where I will test you on something. So before we do that, I want all of you to just quickly revise all the concepts now. So 5 minutes will be okay for all of you. Am I audible? Invisible? Nabil, am I audible and visible? Abia?
Yes, sir. Both, sir.
Yeah. Okay, cool. So just go through the notes quickly and we'll do an in-class assignment right now to see how much you have learned about evolution and what can you infer out of some pictures. Okay. So I think 3-4 minutes will be fine for you to go through, just go through things. And if you have any doubt, please ask before we move forward because that part is crucial. Are you doing, everyone? Minashi?
Yes, sir. Can you scroll up? I don't have the notes.
Okay. Which part you want me to scroll up? Go where?
Yeah, fine. Yes.
Yeah. Okay. So take a few minutes. I'm here. And let me know when you're done. Just write in the chat when you're ready. And we'll start with this assignment. Okay, everyone. Got the information? I'm having some low bandwidth issues for internet. Everyone received a message clearly, right? Yes, no. Nabil, am I audible to you?
Yes, sir.
Okay, cool. Please revise and write in the chat when you're done. Take 2-3 minutes. Are you done, everyone? Am I audible, people?
Yes, sir, you're audible.
Okay. So, is it done? 2 minutes more. Okay, Abhi, I want some more time. Okay. So, I'm giving you a few minutes more, everyone. Then we'll do this assignment. Abhi, please write in the chat when you're done, and others also. I want everyone to be on the same page. Okay, Abhi, can you hear me? Okay. Yes, Minashi, I can scroll up. Just one moment. Which part do you want me to scroll up? Can you write the part here? Is this fine?
Yes, sir.
So I'm waiting for a couple of more minutes, 2-3 more minutes. Okay, everyone, are you done? People, are you done? I'm still waiting. Please confirm when you're done so that we can start. Hope I'm audible. Okay, so let's begin. So what we'll do is this part that we're going to take now is basically in class assignment. So you have to summarize the pictorial evolutionary graph. Okay. So I'll show you a picture. It's from your NCRT only, but the point is this, this picture speaks 1,000 words. As they also, it is always said that diagrams speaks 1,000 words, but these 1,000 words are not written. Okay, but if you can understand the summary of this kind of evolution happening in plants and animals, you will also like get the overall account, the brief account of evolution. Okay, so let me just insert it for you. Minashi, Nabil, and Abia, can you hear me?
Yes, sir.
Okay, now can you see this image? Take it. Make it big. Can you all see this image? Everyone?
Yes, sir.
This is from your NCRT. Is it visible clearly?
Yes, sir. Bryophytes, synapsids, ginkgos.
Yes. Right. So let me explain this image to you, and basically you are going to just write a small summary right now. So first you will write and then I will. So see, on this axis here, this axis, which is your y-axis, it's the time, the era. Okay. So basically time, evolutionary time, or the time period, evolutionary time period. So Paleozoic, Mesozoic, and Cenozoic, we call it. The earliest one is Paleozoic, then Meso means middle one, and Ceno means the current one. And this is where before the Paleozoic, it is a evolution of plant forms, how plants evolve. So chlorophyte ancestors, you can understand the term chloro, right? Tracheophyte ancestors, Rhinia type plants, Xylophyton, and then these things which you can see here. I'm not sure if you can see this branch, but these are branches coming out. They tell you about branching descent, right? Now
Yes,
what you
sir.
have to do is you have to just look at this graph and all the labelings that are there, and you have to write like in one lines at least. So it talks about Silurian, Devonian, Carboniferous, Permian, Triassic, Jurassic, Cretaceous, and then Tertiary and Quaternary. Okay, you have to focus on angiosperms, which are the flowering plants, and you have to focus on mostly on, let's say, these conifers, because some of these plants are present around us. Coniferous plants, dicots and monocots, and write a account. What do you understand? You have to just analyze this graph and tell me. Write important points, and then I'll ask everyone one by one. Okay, do you understand what you have to do?
So like we have to convert this figure into words.
Yes,
Basically.
you have to tell me which, for example, conifers. So take conifers, dicots, and monocots, and if you can take ferns as well. Ferns, don't write about anything else. Ferns and bryophytes, because all these you have studied in class 11, so they can be asked. So you have to tell me when did they emerge and from which ancestor type. Okay, bryophytes, I have made the star and a tick in front of the categories which you have to include.
So we have to show how each angiosperm, how it developed, or we have to show like how the chlorophytes ancestors, then trachophyte ancestors, then these things, then eventually they branched out, or we have to first show their individual cycle, individual evolutionary.
No, you just have to tell that angiosperms, which are the flowering plants of today, both monocots. Let me give you an example of angiosperm and explain it to you. Okay, what you have to do in that today. So you have to write that angiosperms, flowering plants, both monocots and dicots, have evolved from seed ferns. Like their common ancestors are seed ferns, and seed ferns are their common ancestors. Like see, angiosperm is here and cycads are here. So seed ferns is a common ancestor for both cycads and angiosperms. You understand? And seed ferns in turn have evolved from progymnosperms. Understand? Gymnosperms, angiosperms. Cycads are gymnosperms. Make sense to you?
Yes, sir.
And progymnosperms came again from xylophyton. So you just have to connect it till here, because till before that, it's actually understood. Xylophyton, Rhynia type, like this. So now you have to do the same for conifers, ferns, bryophytes. Okay? Do it quickly. I'm giving you 5 minutes, everyone. If anyone has any doubts, please ask in the chat or through the microphone.
Sir, the names are located a bit up and down, sir. Is that indicative of their of which period they started out?
Yes. So this, and yes, and that also you have to take into consideration. So angiosperms come from quaternary, tertiary to quaternary period. Can you see this here?
Yes, sir.
But dicots evolved first. Makes sense, right? Then came monocots. Monocots evolved later. Can you see?
Yes, sir.
Monocots evolved at the same time like bryophytes evolved. Yes, everyone? Yes. Thank you for this point. Maybe. Done, everyone? Did you understood something? Nabil, Minashi, and Abia?
Yes, sir.
Minashi, Abia, could you understand this graph? Minashi, Abia, am I audible?
Yes, sir.
Yes. Are you understanding what this graph is trying to tell you? Okay, so let me ask certain questions. Okay, my first question is for Abia. Abia, which of the current, like the ones that have star marked, like let's say bryophytes, angiosperms, ferns, conifers, and like in angiosperms, both monocots and dicots. Which of the following starred categories do you think has the most primitive features? Which has the most primitive features? Abia. Yes. Am I audible to you, Abia? Minashi?
Yes, sir.
Yeah. Which one of the following, which are star marked here, bryophytes, ferns, conifers, dicots, and monocots? Which one has the most primitive features in evolution? Primitive characteristics. Basically, which one is surviving still with most primitive features?
Ferns.
Ferns? Why do you think it's ferns?
Mm.
Why do you think it's ferns?
I just guessed it.
You don't have to guess, no, the data is in front of you. You will not go and guess in entrances, right?
Yeah.
Yeah, you don't have to guess. Just look at the graph. Nabil, what do you think? Is it there, Nabil?
So the bryophytes,
Bryophytes? Why? Why do you think it's bryophytes?
they have like
Abi also says so in the chat, but yeah, explain. Why do you think it's bryophytes?
they have emerged from the oldest ancestors, namely the chlorophyte ancestors, that emerged directly
Yes,
from and have not branched, gone any more changes since that period.
exactly. So bryophytes have underwent the least branching descent, right?
Yes, sir.
So it's like in animals, when we talk about animals, there are certain animals which are called like very, very close to primitive organisms, right? So this bryophyte, you can see here, this is bryophytes, and it goes here, the least branching, right? Directly to this chlorophyte ancestor. Chlorophyte ancestors evolved or developed, and then before they became tracheophyte ancestors, before tracheophyte ancestors developed from them, a branch diverged to form bryophytes, and it still is a valid plant, right? You understood,
Yes, sir.
everyone?
But relatively,
Yeah.
they've evolved newer as compared to conifers and ferns because they came up during the
Yes.
Jurassic.
Exactly. This is what I was also going to tell. But relatively, bryophytes evolved newer, so they are only seen in. And how do we know that they evolved in Cenozoic only, not in Cretaceous? By fossils, right? We have fossil records of ferns and conifers, okay, even cycads from Jurassic region. Jurassic was the region where dinosaurs were also present, right? Like the reptiles, dinosaurs were present, but bryophytes are not seen. Bryophytes relatively evolved later. Is this clear
Yes, sir.
to Minashi and Abia? Are you also understanding what this exercise was meant to? Yes, this is how you have to read it.
No,
Okay.
so which
Now,
is older, sir? Ferns, conifers, or bryophytes?
sorry, which is more
Older, sir. Like,
older?
yes, sir.
Yes, so ferns and conifers, they both are very old. Like if you say older, so it's not like all ferns that we see today around us came only in the Jurassic. There were ferns already. Now ferns also have speciation. They also have undergone speciation. So bryophytes, the first kind of bryophytes, which could have been called as bryophytes, they evolved late. So from here to here also in tertiary and quaternary also there are many species that have now come up, right? So ferns also, the ferns which are present today, like here, they are very different from the ferns in terms of they are same also, but in size, in habitat, niche, and other things, there are many species that have come, but they're all fern species. It's like there are many cats on the planet, right? Tiger, leopards, cheetah, lion, jaguar, what else? All these, they are all cats and are normal cats, Siberian cats. They're all cats, right? So ferns and conifers are oldest among the three. Do you understand, Nabil?
Yes, sir.
This period, like from here to here, and for both like ferns and conifers, it also tells that there were organisms, there were plant species that were evolving and developing and adapting, you know. But the closest that ferns that we have, basically, it started from here only, Devonian. So there were some organisms here that looked like fern. They kept on evolving. It's like that. Do you understand? But they majorly flourished and took over in this period, Jurassic. Do you understand this graph,
Yes, sir.
Nabil? Okay, very well. Everyone, it's clear, right? Now, write down certain points before we move forward. Write down a brief account of evolution. A brief account of evolution. First, we'll talk about plant evolution. Write down. Okay, before plant, sorry, first, before plant evolution, write down some general statements. The first form of life. Now your NCRT says, if you have NCRT, all of you have NCRT, can you open? And can you confirm also, because in my opinion, like if I'm not wrong, the NCRT states that first form of life evolved somewhere around 2 billion years ago. If can someone confirm?
3 billion, sir.
Yes, it is. It's in fact, it's the evidences now suggest that it is even more than 3. It's around 3.5 billion years old. But your NCRT states it's 2 billion years old. Okay, so you have to stick to that for entrances, but you should know the truth. Like it's outdated and information. Is it clear?
So
Clear,
I think it's
Minash?
in NCRT also. I think it's 3 billion.
Is
I
it?
think.
Okay,
Yes,
can you
sir.
please confirm?
1 minute.
Yeah, please open the NCRT and you can find it. If it is 3, then it's very good, because it's actually in reality more than 3, but 3 will be about 3 to 3.2 billion years ago. The first cellular form of life evolved.
No, sir, it's 2 billion.
It's 2, right?
Yes, sir. But in
Because
the starting, the first topic they've given about 4.5 to 3, but here they've given suddenly 2 billion.
yeah, so that's actually not Correct. So write down the first cellular forms of life. The first cellular forms of life the first cellular forms of life evolved around 3.2 billion years ago. BYA means billions years ago. MYA means million years ago. Okay. Just notice. And then write down non-cellular aggregates. As I've told you, we are just summarizing everything. Non-cellular aggregates of molecules. And write down inorganic, of inorganic molecules assembled into membranous cells with membranous coverings or envelope. Okay, so this happened. And to tell you that when life evolved, remember that what did Operin, Haldane proposed, and what did Miller also proved? That the atmosphere at that time was reducing atmosphere, right?
Yes, sir.
Yes. What do we mean by reducing atmosphere?
They had hydrogen, less oxygen, more hydrogen, and
Yes, the oxygen was not present actually. Like that is what is believed. Now, though there are many recent studies that argue about how much oxygen was available and how much was the environment really that reducing. Was it a little bit oxidizing? Because if it is little bit oxidizing, that helps to explain the evolution of life better and things like that. But majorly, it was lacking oxygen. Okay, is it clear? Majorly it was reduced. That's the current understanding. But then certain cells, like certain simpler organisms, like unicellular photosynthesizing bacteria and algae, started evolving. Is it clear? And when they started evolving, they started producing oxygen, because one byproduct of photosynthesis is what? What is the byproduct of photosynthesis? Everyone. Oxygen. What is released during photosynthesis? Am I audible or not audible? Okay. So oxygen is evolved during photosynthesis. So write down some of the early organisms evolved, some early unicellular organisms evolved the ability to release oxygen. To release oxygen and start making the environment less reducing. You know, and interestingly, let me tell you, when oxygen first started coming up in the atmosphere, okay, is oxygen a good thing for a cell or a bad thing for the cell? What do you think? Or is it neutral? Nabil, Minashi, am I audible? Yes, someone is
Yes,
saying
yes.
something. We are saying neutral. Okay. So you cannot say it without knowing the concentration. Okay. So at lower concentrations, if an organism is aerobic, less oxygen is harmful for that organism, like humans. But also at very high concentrations, oxygen is very, very detrimental. It is very, very damaging to each and every cell. So high concentration of oxygen also, because you know, oxygen, one thing it is good at is breakdown. It breaks, it oxidizes molecules. So combustion is nothing but just oxidation, right? When we burn something, burning involves oxygen, right? You cannot burn without oxygen. Do you know this? Yes or no?
Yes,
We'll,
sir.
yeah, right. So when oxygen started coming into the atmosphere early on, there was lots of oxygen toxicity for early evolving organisms, and that's where aerobic organisms started evolving, which can tolerate oxygen and use the oxygen for their own benefit to oxidize glucose. Before that, it was believed that for to create energy, something else apart from oxygen was being utilized. Okay, when oxygen came, then it was a more suitable uh, element that can be used. Okay. Okay. So okay, write down few more points. Let's just finish this part. So oxygen started coming into the environment and started making the environment less reducing. And so these early organisms which could release oxygen, these early organisms which could release oxygen had evolved chemical reaction pathways, had evolved chemical reaction pathways similar to photosynthesis. Similar to photosynthesis. Is it clear?
Sir, so first organism, why not photosynthetic?
No, because first organisms were not photosynthetic. They were chemosynthetic. Remember, bacteria is chemosynthetic? Mostly they were just utilizing chemicals around them, right?
Yes, sir.
Yeah. Monera, kingdom Monera.
Sir, and I've heard somewhere that now we have plants that have chlorophyll. Earlier they had like a reddish pink pigment instead of chlorophyll.
Yes. Even chlorophyll comes with like chlorophyll A and chlorophyll B. There is a chlorophyll C and other type of related pigments, xanthophylls, anthocyanins. These things are also there. Chlorophyll just happen to be the blue-green pigment basically. But other pigments like carotenoids, xanthophylls, they are also in other color, like they are reddish, brownish pigments. So chlorophyll is the most uh, suitable, like it's the most efficient pigment that actually can help in the process of photosynthesis. It's not like non-color pigments do not do photosynthesis, but the rate of photosynthesis is very, very less. They cannot do it with the like in the most efficient way from the light energy from the sun. Okay. So earlier when plants evolved, also because it is believed that the concentration of oxygen availability, the concentration of carbon dioxide which was available in the atmosphere, they were all very different in early Earth, right? Today, whatever is the atmospheric constituent composition is fixed from last 3 million years ago. It's fixed. It's like that only. 21% oxygen, 78% nitrogen, 0.3% carbon dioxide, and trace gases less than 1% water, etc. Do you understand, Nabil?
Yes, sir.
Yeah. So we know that early organisms that evolved ability to release oxygen could do something like photosynthesis. It was not exactly photosynthesis, but something like the light reaction. And in the photosynthesis also, which part? The light part, the light reaction of photosynthesis. And then we'll come to animals will do tomorrow. Okay, so let me stop here. And tomorrow I'll do animals, and the same kind of activity you will do tomorrow with animals. So I urge you all to go back and read the last segment of your NCERT for evolution chapter, where they have talked about animal evolution. So tomorrow we'll finish this chapter, we'll do animal evolution, and then finally we'll come to human evolution, which will take around half an hour in itself. Okay, so how did humans evolve?